We are actually looking at turtle stool samples for the seeds and the plants that they contain. So we have two different samples. One was obtained at a height of 206 meters above sea level, and then the other one was obtained at 60 meters above sea level. So we're looking at the different compositions of plants that grow there, because by looking at what the tortoises are eating, you can actually figure out what sort of plants are growing there, and then also if the tortoises are migrating, as they do, what sort of seeds are being carried to different environments, and then what seeds are thriving in those environments versus the ones that are not. For example, the tortoises really like to eat guava fruits. Guava is actually an invasive species here on the Galapagos. So what can happen is a tortoise can eat a guava tree from a higher elevation and then carry it to a new environment where then it can outcompete. There is a native guava tree, however they only have 4 seeds per fruit as compared to the 600 seeds per fruit of invasive guava. So already, just as the number of offspring, they're being outcompeted. The invasive guava trees are much larger than the native guava trees. So what can happen is they're also outcompeted for light as well. So when we were preparing the stool samples to look at the seeds, I remember thinking this is similar to, but way better than the owl pellet dissection that's traditionally done in biology classes. I think that it has a little bit more instructive value. This has instructive value as to what sort of species not only exist in that area, but also how they can go from one area to another through the host species. So I think that's very interesting to look at species distribution as well. In the classroom, students kind of expect there to be, we're done in 15 minutes, we follow this like set lab procedure, we get this set result, and ta-da, you have science. Here out in the field, it's not necessarily like that. Like, it took us a while to find an acceptable sample, instead of just having it prepared like with the owl pellets, like ta-da, owl pellets. But here we actually had to go look for it, and it's not a guarantee. So I think that's the big difference in that, science in the classroom is neat, guaranteed, it's prepackaged, versus science out in the real world. The data can be really messy, it can be unexpected. And it also just requires not only a lot of hands-on work, but a very much minds-on sort of approach as well.